Hello, I'm Mridul Mohan from group number 324. So today's topic is cell membrane, the first topic. Cell membranes from a biophysical perspective. What's cell membrane? Cell membranes are the dynamic and essential boundaries that define and protect cells. They are composed primarily of lipids and proteins, forming a complex and intricate structure that regulates the exchange of molecules between the cell and its environment. It is easy. It's in this we will delve into biophysical properties of cell membranes, exploring their structure, function, and underlying physical principles, and govern their behavior. So, the first come the structure of cell membranes. The fundamental building blocks of a cell membranes are phospholipids, amphipathic molecules, and a hydrophilic head and a hydrophobic tail too. These phospholipids spontaneously assemble into a bilayer with their hydrophobic heads facing the aqueous environment on either side and their hydrophobic tails forming a hydrophobic core. This bilayer structure provides the basic framework of the membrane, creating selectively permeable barrier that separates the intracellular and extracellular compartments. The lipid bilayer, the second layer. The lipid bilayer is not a static structure but a dynamic fluid mosaic. The individual phospholipids can move laterally within the plane of membranes, allowing the fluidity and the flexibility. The fluidity is essential for various cellular processes, including cell signaling, membrane trafficking, and cell division. Membrane proteins. Proteins are embedded within the lipid bilayer, performing a variety of functions. Integral membrane proteins span the entire membrane, while peripheral membrane proteins are associated with the membrane surface. These proteins play a crucial role in transport, facilitating the movement of molecules across the membrane through channels, carriers, and pumps. Signaling, receiving and transmitting signals from the extracellular environment. Cell adhesion. This is a very crucial topic, connecting the cells to each other to the extracellular matrix. Enzymatic activity catalyzing biochemical reactions. So, biophysical properties of cell membranes. Fluid mosaic model, a very important model. The fluid mosaic model describes the dynamic nature of cell membranes. The lipid bilayer is fluid, allowing for a lateral movement of lipids and proteins. The fluidity is influenced by factors such as temperature, lipid composition, and cholesterol content. Cholesterol, for example, can modulate membrane fluidity by interacting with phospholipids and reducing their movement. Membrane potential. The difference in electrical charge across the cell membrane is known as the membrane potential. This potential is generated by the unequal distribution of ions, primarily sodium, Na+, and potassium, across the membrane. The potassium and sodium pump actively transports sodium out of the cell and potassium of the cell, creating a concentration gradient. This gradient, along with the selectively permeability of the membrane of different ions, contribute to the resisting membrane potential. Membrane transport. Cell membranes regulate the movement of molecules across the membrane through various mechanisms. So the first mechanism is passive transport. These transport we are learning from from class 8, 9, yeah. Diffusion of molecules down the concentration gradient without the expendation of energy. This includes simple diffusion, facilitated diffusion, and osmosis. And active transport. Movement of molecules against their concentration gradient requiring energy, input of ATP hydrolysis. This is carried out by the protein pumps such as sodium and potassium pump. Membrane fusion and fission. Cell membranes can undergo fusion and fission processes, allowing for exchange of material between the cells or the formation of new organelles. These processes involve complex interactions between the lipid and proteins, regulated by specific signaling pathways. Membrane curvature. The curvature of the cell membranes plays a crucial role in various cellular processes such as cell division, vesicle trafficking, and organelle formation. Membrane curvature is influenced by the lipid composition, protein interactions, and cytoskeletal forces. Biophysical techniques for studying cell membranes. A variety of biophysical techniques are used to study the structure and function of cell membranes. So, the first is fluorescence microscopy, visualizing membrane, proteins, and lipids using fluorescent probes. Electron microscopy, high resolution images of membrane structure. X-ray diffraction, determining the arrangement of lipids and proteins in the membrane. Nuclear magnetic resonance, also known as NMR, spectroscopy, analyzing the structure and dynamics of membrane components. Patch clamp, electrophysiology, measuring the electrical activity of ion channels and other membrane proteins. So, in conclusion, the cell membranes are complex dynamic structures that play a vital role in cellular function. Their biophysical properties, including fluidity, membranes, potential, and transport mechanisms, are essential for maintaining cellular homeostasis and enabling various cellular processes. By understanding the underlying physical principles that govern membrane behavior, we can gain valuable insights into it. So, thank you so much. So, I'm starting with the second topic, the axon and the nerve impulse. So, in introduction, the axon, a long slender projection of a nerve cell, a neuron, is primary conduit for transmitting electrical impulses or action potentials within the nervous system. These action potentials are the fundamental units of neural communication, allowing for rapid and efficient information transfer between neurons, muscles, and glands. So in this, we'll discuss about the structure, function, and biophysical mechanisms underlying axon and nerve impulse conduction. So the first one is structure of axon. The axon arises from the cell body at a specialized region called axon hillock. It is typically cylindrical shape and can vary in length from a few micrometers to several meters depending on the types of neuron. The axon often covered by a myelin sheath, a fatty substance that insulates the axon and increases the speed of nerve impulse conduction. So, what's myelin sheath? The myelin sheath is formed by specialized glial cells, oligodendrocytes in the central nervous system, and Schwann cells in the peripheral nervous system. These cells wrap their membrane around the axon, forming multiple layers of lipid-rich membrane. The myelin sheath is interrupted at regular intervals by nodes of Ranvier, which are the gaps in the where the axon membrane is exposed. The second one is the nerve impulse, the action potential, also known as action potential. The action potential is a rapid transient change in the electrical potential across the axon membrane. It is initiated when a neuron receives a stimulus that exceeds a certain threshold. The following steps describe the generation and propagation of an action potential. So, listen carefully. The first is resting potential. A rest, the inside of the axons negatively charged relative to the outside due to the unequal distribution of ions. This difference in the change is called resting potential. The primary ions involve the potassium K plus and sodium Na plus. The resting potential is primarily maintained by selective permeability of the membrane to these ions and activity of the sodium potassium pump. So the second process is depolarization. When a neuron is stimulated, ion channels in the axon membrane open, allowing Na plus ions to flow into the cell. The influx of positive ions causes the membrane potential to become less negative, a process which is also known as depolarization. And the third one is the action potential threshold. If the depolarization reaches a certain threshold, an action potential is triggered. The threshold is minimum level of depolarization required to initiate an action potential. Rising phase, the fourth process. Once the threshold is reached, voltage gated Na plus ions open rapidly, causing a massive influx of Na plus ions. These influx further depolarizes the membrane, leading to a rapid rise in the membrane potential. So the fifth one is the peak. The membrane potential reaches a peak value as an influx of Na plus ions continues. The sixth one is falling phase. Voltage gated K plus channel open, allowing K plus ions to flow out the cell. These influx of positive ions repolarizes the membrane, bringing the potential back towards the resting level. Undershoot, the seventh process, the membrane potential briefly becomes more negative than the resting potential due to the continued influx of K plus ions. This is also known as the undershoot or hypolarization phase. The eighth process is refractory period. During the refractory period, the neuron cannot generate another action potential, ensuring the impulse travels in the direction only. So that's over. And the next topic is propagation of action potential. How it propagates. So the action potential propagates along the axon as a wave of depolarization by unmyelinated axons. The action potential spreads passively along the membrane, causing adjacent regions to depolarize and generate their own action potential. This process continues along the axon, allowing the impulse to travel from cell body to the axon terminal. So in myelinated axons, what happens? The action potential jumps from the one node of Ranvi to the next, a process known as saltatory conduction. This significantly increases the speed of conduction, allowing a rapid transmission of nerve impulses. So, what are the factors that affects nerve impulse conduction? So, the several factors that can influence the speed and efficiency of nerve impulse conduction are the first one is myelination. Of course, myelination. The presence of myelin sheath significantly increases the speed of conduction. The axon diameter. Larger axons conduct impulses faster than smaller axons. The third one is temperature. Higher temperatures increases the speed of conduction, while lower temperature decreases it. And the next is ion concentrations. Changes in the concentration ions such as the Na plus and K plus can affect the resting potential and the ability to generate action potential. Drugs and toxins. Certain drugs and toxins can interfere with ion channel function, altering nerve impulse conduction. So, in conclusion, the axon and the nerve impulse are essential for the functioning of nervous systems. Understanding the biophysical mechanisms underlying nerve impulses conduction is crucial for comprehending the working of the nervous system and developing treatment for the neurological disorders. So, thank you so much for listening.